In the previous session, we discussed about one carbon metabolism pertaining to the definition, how the one carbon groups are generated and interconverted. Okay, in this session, we will be discussing about utilization of one carbon groups. Objective of this session is, at the end of the session, the learner shall be able to describe utilization of one carbon groups. So we talked about the CO2 in earlier session. We said it is not included in one carbon pool. It is dealt by thymine, pyridoxine, and biotin. Okay. Then other than, so we say other than CO2, so they are these one other than CO2, the one carbon groups, these one carbon groups are carried by, these one carbon groups are carried by tetrahydrofolic acid, we say THFA. So we have also shown how they are interconverted one form of coenzyme to the other form of the coenzyme. So these one carbon groups used for synthesis of so here I will mention various one. Okay, the first one we talked about, so glycine, synthesis of glycine. The second one is serine. The third one is so thymine monophosphate, thymidylate. Okay, thymine monophosphate or thymidylate. Okay, then the next one is purine basis. The purine basis. Okay, the next one is methionine, synthesis of methionine. Okay, the next one is so methyl groups carried out by methyl groups carried by S-adenosyl methionine. Okay, S-adenosyl methionine. Where it is donating those methyl groups. Okay, then we'll study one by one. The first one is the synthesis of glycine. Here it is synthesis of glycine. So as we said, so glycine cleavage system is there. So here we have CO2 plus NH4. Okay, CO2 plus NH4. In the presence of glycine cleavage system, we discussed about that aspect. Glycine cleavage system. Here in the glycine cleavage system, as I mentioned, this is a reversible reaction. Okay, here what happens in this one? The N5, N10 methylene THFA is converted to THFA, leading to the formation of glycine. Here, the leading to the formation of glycine. Okay, glycine. Okay, here leading to the formation of glycine. So this is about how glycine synthesis is taking place. In the second reaction, okay, synthesis of serine. It is synthesis of serine from glycine. Here we say, okay, this is so glycine. This is what we say is so glycine synthesis of, okay, so serine from the glycine. Okay, glycine. Here, so the enzyme, it is also reversible reaction. So we can say the SHMT. Here it is. The enzyme is SHMT. Okay, serine hydroxymethyl transferase. Again, we have here N5, N10 methylene THFA is converted to THFA, leads to formation of, okay, the serine. Okay, leads to formation of serine. Okay, So these two reactions we have studied when the folate, the one carbon groups are generated. The third one is we explain synthesis of thymidylate. Synthesis of thymidylate, thymine monophosphate. Okay, here it is synthesized from the uridylate. Another pyrimidine nucleotide, it is synthesized from the uridylate. Okay, let us see here. Okay, here what happens? Generally, uracil carries when it becomes nucleoside, later nucleotide, ribose. Here it is presence deoxyribose is present. So that is the reason we call it deoxyuridylate. We call it deoxyuridylate. This deoxyuridylate, okay, then in the presence of thymidylate synthase, okay, that is thymidylate synthase, okay, thymidylate synthase, then what happens? In the presence of N5, N10, methylene, it is called methylene THFA, N5, N10 methylene THFA. THFA formation is taking place, leads to formation of thymidylate. So here, so we say deoxythymidylate. Okay, then here it is, as you know, that is simply we can also call thymidylate. Okay, that is as thymine always with the deoxyribose. See the difference here. See the difference here. Here suppose here H is present here. Okay, then see here the thymine, that is methyl group, is transferred here. You are done. So fifth position. Okay, then this is what we call thymidylate. So in fact, so here it is as a nucleotide, it will have the phosphate also. Here I say it will have the phosphate. Okay, so simply we can say TMP. The next reaction is, that is synthesis of purine bases. So some of the groups of purines so is contributed by these one carbon groups. So let us see here. So let us see the structure of purine ring. Okay, let us see the structure of purine ring. Okay, so here, okay, here it is. Okay, so in this the purine ring, here it is. Yeah. Okay, here it is. Okay, so let me carry out the numbering of, okay, purine ring. Okay, then carry out the numbering of the purine ring. Okay, then let us concentrate on this aspect. Here, what is that one? Here it is. Okay, so let me concentrate on this aspect and also here. Okay, so here what happens? To this, here both, when you talk about both, so this is provided by N10 formyl THFA, donated by N10 formyl THFA. This is donated by N5 N10 methynyl THFA. So there are some issues about this particular one. So when you talk about this particular aspect, so there are different books mentioned about the source of this C8. Some of the books it mentioned N10 formyl THFA. Some of the books it mentioned N5 N10 methynyl THFA. So that this one I have taken as a reference from the Harper, where it is mentioned N5 N10 methynyl THFA. Okay, then the next aspect is, the next one is N5 formyl THFA. What is the role of this N5 formyl THFA? So it does not transfer, not involved in the, it does not transfer one carbon groups. So if it doesn't transfer one carbon groups, okay, then what is its use? It has got the uses in the supplementation. What is it? It is also called form factor, citroform factor, okay, or folinic acid, folinic acid, folinic acid. Okay, this is used as supplement to folic acid. Why it is used as a supplement to folic acid? So there are two uses are there. The first one is, so it can be converted so to other forms, can be converted to other forms. Okay, so that is the one use of this one. The second use is more effective. It is more effective, okay, than folic acid. More effective than folic acid. It is more effective than folic acid. So this, because of these two reasons, so it is used as a supplement. The next one, fifth one, is the synthesis of methionine. Synthesis of, okay, methionine from homocysteine. It is synthesis of methionine from homocysteine. Okay, let me write the structure of homocysteine. Okay, here this is the structure of homocysteine. Okay, this is homocysteine. Okay, the homocysteine. So in the presence of homocysteine, methionine, methyl transferase, homocysteine, methionine, methyl transferase. Okay, in the presence of B12, this reaction can only take place in the presence of B12. The reason is N5-methyl THFA is converted to THFA. Okay, leads to formation of methionine. Okay, here this is methionine. Okay, then formation of methionine. So this is one of the very important reaction where the folate coenzymes are involved. Okay, in this what happens? As I said, formation of N5-methyl THFA is an irreversible reaction that was discussed in the previous session. Okay, here the N5, okay, methyl THFA, N5-methyl THFA can only transfer, okay, so methyl group to cobalamin. It is cobalamin. So converting it into methyl cobalamin. So it is transferring to the B12. So that is cobalamin. Converting into methyl cobalamin. So okay. So what happens? This methyl cobalamin, what it does? It transfers that methyl group to okay, homocysteine, okay, converting it into methionine. Okay, that means without the B12, this methyl transfer cannot take place. That means N5-methyl THFA so cannot be converted to THFA in the absence of B12. That is the reason in the absence of B12, the folate is trapped in the form of N5-methyl THFA. That is the reason whenever the folate is given, B12 has to be given if it has to be utilized properly. The methionine, okay, leads to formation of S-adenosyl methionine. Okay, methionine, another active form. So this can donate the methyl groups, leads to formation of choline, and leads to formation of epinephrine from norepinephrine. Okay, and leads to formation of carnitine, okay, then leads to formation of, so there is transfer, methyl groups transfer RNA and also DNA, etc. Okay. Then the importance, that is the one importance. General importance of one carbon metabolism is, what is the importance? Okay, they are at the different level of oxidation. They are transferred, okay, through what? Mostly through what? Through THFA, and vitamin B12 also used, okay. Then mainly useful, okay, in anabolic reactions, that is synthetic reactions. So as you have seen, formation of choline, formation of epinephrine, formation of carnitine, etc. They have very important role in the metabolism. Also, there is transfer RNA and DNA. Let me summarize one carbon metabolism. Part 2, utilization of one carbon groups. Objective: at the end of the session, the learner shall be able to describe utilization of one carbon groups. So apart from the CO2, other one carbon groups are carried by THFA, used for the synthesis of glycine, serine, TMP. Okay, then purine bases, methionine, and methionine in the active form, S-adenosine methionine, transfers the methyl groups. Then the post-mortem synthesis of glycine from the CO to NH4, glycine cleavage system, N5N10 methylene THFA converted to THFA, leading to formation of glycine. The next reaction, synthesis of serine from the glycine by the serine hydroxymethyl transferase, N5N10 methylene THFA leads to THFA, leads to serine. Okay, then synthesis of thymidylate from the deoxyuridylate. So by the thymidylate synthase, methylene THFA converted to thymidylate and TMP. Then synthesis of purine bases. Okay, then here, so C2 of the purine and C8 of the purine, C2 is supplied by N10 formal THFA, C2 is supplied by N10 formal THFA, and C8 is supplied by N5 N10 methynyl THFA. Then we talked about the N5 formal THFA does not transfer one carbon groups. It is called citrovarum factor or folinic acid, used as a supplement to folic acid. It can be converted to other forms easily, more effective than folinic acid. That is the reason it is used as a supplement. Then synthesis of methionine, where from the homocysteine by the action of N5 methyl THFA in the presence of enzyme homocysteine methionine, okay, then methyl transferase, and leads to formation of methionine. Here, as I said, N5-methyl THFA formation is a reversible reaction. This can only transfer methyl group to the homocysteine in the presence of B12. That is the reason in the deficiency of the B12, folic acid deficiency also will be there, because folate is not available. It is trapped in the form of methyl folate. Okay, then, so what happens, how it takes place? N5-methyl THFA can only transfer the methyl group to cobalamin, leading to the conversion of methyl cobalamin that transfers the methyl group to the homocysteine, leading to formation of methionine. Then, so the methionine can be converted to active form S-adenosine methionine. It transfers to the choline, leading to the formation of epinephrine from the norepinephrine, carnitine, and also transfer the methyl group to the transfer RNA, DNA, etc. The importance of one carbon metabolism is these groups are transferred through the mostly THFA, and in one reaction is B12 leads to many synthetic or anabolic reactions. With this, we have completed the one carbon metabolism. Thank you very much.